Hello students, in our yesterday's class we were discussing about nucleic acids. So in case of higher animals including lower animals or the organisms including viruses, we will be having two types of nucleic acids. One will be DNA and other one will be RNA, as you know that. So when we write DNA and RNA, so yesterday I told you what is the fundamental difference between DNA and RNA. That is, at second carbon atom we will be having OH group here, and in case of DNA only H is present. I mean to say that O is being absent. So that if O is being absent means we need to use the word deoxy. So remaining ribonucleic acid as it is. So that DNA deoxyribonucleic acid. So this is the fundamental difference, and still many difference are there. So that can be discussed in further classes later. So here we discussed about, that is main important, the composition. I mean to say the chemical composition of DNA and RNA. I mean what are the different chemicals present in the DNA and RNA. So one is, that is sugar. We can call it as pentose sugar. So also called as ribose sugar. I told you yesterday, ribose sugar. So structure will be like this. Here, H, here will be having OH, CH2, OH, 1, 2, 3, 4 and 5. So there are 5 carbon atoms are there, hence it is called as pentose sugar. Clear? So this is first the chemical composition or the ingredient present in the nucleic acid. Second one, nitrogenous bases. Again there are two types. One will be purines, another one is pyrimidines. So in the purines again there are two. One is adenine, second one is guanine. So represented as A, represented as G. And next again pyrimidines are of two types. One is cytosine and thymine. Cytosine, thymine. In case of RNA we will be having, I mean instead of thymine we will be having uracil. Do remember. So here C represented as then T and uracil U. This is how we can represent, and these two are, I told you yesterday, double ringed, and these three are single ringed. That is, here you will be having like this. Clear? So this is what? Second one, nitrogenous bases. And last one, that is phosphoric acid. It is in the form of phosphate, that is HPO4. This is what? The third ingredient or the chemical composition present in the nucleic acid. And by understanding all this, I mean to say that when you take a DNA or the nucleic acid, how these three are arranged, in what fashion they are arranged. So that can be discussed in today's class. So first one is nucleotides. I mean to say that what is DNA or RNA? Very simple. Long chain of nucleotides. Long chain of nucleotides. I told you yesterday, long chain of amino acids is called as protein. A long chain of fatty acids is called as lipid. Likewise, long chain of nucleotides is called as DNA or RNA. This is what? See. And how the nucleotides are formed. So before understanding this, we need to understand nucleosides. Nucleosides. How the nucleosides are arranged. So that is important. And here, very simple formula. Pentose sugar. Do you remember what mean by nucleosides? Pentose sugar combines with any one of the nitrogenous base. Very simple formula. That is, nucleosides means when pentose sugar combines with any one of the nitrogenous bases which gives or which yield, that is nucleoside. Very clear? We'll take a particular, that is there are 5 types of nucleosides are there. We'll discuss one by one. Pentose sugar, as it is pentose sugar. Next, instead of nitrogenous base, that is a common, and here we'll be having particular, so that instead of nitrogenous base we'll take one nitrogenous base, that is adenine. Clear this? So when pentose sugar combines with the one of the nitrogenous base, that is particularly adenine, so it will become what? Nucleoside. Here adenosine. Clear that? Adenosine. So now the adenosine is considered as one of the nucleoside. This is what, how to write the nucleosides. Very simple formula. Pentose sugar combines with any one of the nitrogenous base which gives nucleoside. When you take a particular pentose sugar, I mean to say this structure, here you will be having any one of the nitrogenous base at first carbon atom of pentose sugar. Now this structure is considered as nucleoside. Very clear? Next, second one. Same, pentose sugar, instead of adenine I can put guanine. Yes, can you imagine what would be the nucleoside? Yes, think in your mind, what would be the nucleoside? Definitely that would be guanosine. That could be guanosine. Very simple. And third one, again same, pentose sugar, instead of guanine I am going to put cytosine. Definitely it yields, that nucleoside is considered as cytidine. Cytidine. See, next again one more, pentose sugar plus a thymine which gives thymidine. Thymidine. Next, instead of uracil, if I put uracil, it will become uridine. Clear? So adenosine, guanosine, cytidine, thymidine, uridine are considered as nucleosides. I mean to say these all 5 are considered as nucleosides. Hope you understand. Next. Then here first we need to understand nucleoside. So simple formula is pentose sugar plus any one of the nitrogenous base over. When we take nucleosides, we can form nucleotides. Now we start writing nucleotides. Nucleotides. Again there are 5. Very simple formula. The nucleotides means pentose sugar plus any one of the nitrogenous base. Simple. See, pentose sugar plus any one of the nitrogenous base which gives nucleoside. Yes or no? Which gives nucleoside. So now nucleoside plus HPO4, we can call it as phosphate. Clear? We can write phosphate, that is HPO4. It will become nucleotide. Simple formula. So I mean to say that for this formula, pentose sugar plus nitrogenous base which gives nucleoside, nucleoside plus HPO4 which gives nucleotide. Simple? Yes, we start writing. See, first one here, nucleoside plus HPO4 which yield nucleotide. Yes. Then we will take very specific. See, pentose sugar plus adenine which yield adenosine, nothing but nucleoside. So to this nucleoside I am going to write HPO4, that is phosphate. Yes, now it will become, it will become nucleotide. That nucleotide is adenylic acid. Clear? Adenylic acid. So that adenylic acid is nothing but nucleotide. This is one of the nucleotide. Adenosine, nucleoside. Adenylic acid is a nucleotide. Very simple. Second one, pentose sugar plus guanine. which gives guanosine, nothing but nucleoside. Nucleoside plus HPO4, which gives, yes, think in your mind. Think in your mind. Yes, it will become adenosine, adenylic acid, guanosine, guanylic acid. Guanylic acid. This is what? Second what? Nucleotide. Then third one is cytidine. Cytidine plus HPO4, which yield, that is nucleotide, that is cytidilic acid. Nucleotide is cytidilic acid. So a little bit confusing words. Again plus HPO4, that is pentose sugar plus thymine, thymidine. Thymidine plus HPO4, which yield thymidilic acid. Next, last one, uridine plus HPO4, which gives, that is uridilic acid. Is that clear? So these are the 5 nucleosides and these 5 are nucleotides. Clear? So I can show this structure. Yes, this is what? First one, pentose sugar. I can write here pentose sugar plus any one of the nitrogenous base. Here I can write what? Nitrogenous base. Now the structure become nucleoside. Clear? So here instead of nitrogenous base you can write adenine, you can write guanine, you can write cytosine, thymine, whatever. Clear? It will become adenosine. Same time at third carbon atom, I am going to put HPO4, that is phosphoric acid. Now it will become nucleotide. I mean to say this is what? Adenosine. Adenosine plus phosphate, it will become adenylic acid. Nothing but nucleotide. So this is one nucleotide. Again you will be having, yes, one more structure called as pentose sugar. Again you will be having one more nitrogenous base, again HPO4, and here you will be having CH2OH here. Clear? So now one nucleotide, second nucleotide, third, 4, 5, 6, like this. I mean to say long chain of nucleotides is called as nucleic acid, that is DNA or RNA. Very simple to understand. Yes, definitely in the examination they are going to ask. Very simple to write. See what mean by DNA or RNA, what is the fundamental difference and where they are present, and what are the nucleosides and what are the nucleotides. So this is how to write in the examination. Yes, we'll move on to the next topic. Yes, after this nucleotides or nucleic acids, we will have one more topic, that is primary and secondary yes. Now we have primary and secondary metabolites. Primary and secondary metabolites. So very simple and small topic. So primary and secondary, the next word is very important, simple word is metabolites. What mean by metabolites? What are metabolites? Where they are present? Who can prepare or who can produce these metabolites? Yes or no? So this whole concept is come under primary and secondary metabolites. So very simple to understand. When you take, I mean to say that when you take a plant, in the plant you will be having different tissues such as leaf, stem, root, flower, bud, whatever you take, so that contain tissues. Yes or no? So simply called as plant tissues. plant tissues. So that when you take plant tissues, you will be having several thousands of or several different types of chemicals or chemical compounds. As you know that is already we conducted an experiment, that is what? Chemical analysis, and second one is destructive experiment. So by means of those two experiments, definitely we categorized several thousands of organic compounds, inorganic elements, inorganic molecules and inorganic compounds. Yes or no? So that when you take a plant and or plant tissue, so when you conduct an experiment by taking the sample of plant tissues, we will be having several thousands of different chemical compositions. Yes or no? So in that, some of the chemicals are directly involved in the what? Normal body physiology or normal physiology of the plant body, growth of the plant body, and what? Synthesis of phytohormones or plant hormones. Yes or no? So many different phenomena are there, like flowering, that we can call it as vernalization, and seedling. Yes or no? Sorry, seed formation, flower formation. Yes or no? Bud formation, leaf formation, root elongation, stem elongation. Many different phenomena are there in the plant body. So each and every phenomenon are going to take place in presence of a particular chemical. That is, say for example, the closing and opening of stomata is taking place under the influence of potassium, that is K plus. So that each and every chemical, so they are going to play their respective role in the plant body. So for that reason, the plants contain several thousands of different types of the chemical compounds in their body. So that here, out of several thousands of chemicals, how can we categorize into primary and secondary metabolites? No doubt, whatever the things are going to produced by the plants as their, what we can call it as byproduct. The better word is byproduct. So the metabolites means byproduct. Best word is byproduct. I mean to say that during their growth, during vernalization, I mean to say flowering, during bud formation, root formation, or whatever, so those plants are able to produce some of the different chemicals. So those chemicals nothing but the byproducts. So those all byproducts nothing but metabolites. Clear my point? Whatever the materials or the chemicals are produced by the plant during their physiology is considered as metabolites. In that, how can we categorize? What are the different strategies to understand which one belongs to the primary metabolites and which belongs to the secondary metabolites? That is our today's topic. Very simple. That is, the plants contain, as you can understand, sugar. Yes or no? Sugar, we can call it as polysaccharides. Sugar nothing but polysaccharides, or better word you can call it as carbohydrates. Second one, protein. Next one is lipid, and fourth one is nucleic acids. So these four are simply considered as primary metabolites. Do remember. So these four are simply considered as primary metabolites. Now you can ask a question, sir, how? Based on what strategy you are telling these four belongs to the primary metabolites? Yes, very simple technique to understand is, so those chemical compounds or those chemicals which are synthesized by the plant body and those are directly involved in the normal physiology. Normal physiology plus growth plus development. Clear my point? So first one is normal body physiology, growth, development, and different other phenomena, as I told you right now, vernalization, that is flowering, seed formation, fruit formation, etc. So those chemicals directly involved in these processes are considered as primary metabolites. Very simple. Yes, those chemicals synthesized by the plant body which are directly involved. Please note it, write down. So those chemical compounds which are synthesized by the plants, not only plants, it may be plants, not only plants plus fungus plus microbes. Clear? Microbes. So those chemical compounds which are synthesized by the plants, fungus and microbes, which are directly involved in the normal physiology, growth, development and etc. processes, are simply considered as primary metabolites. This is first one. Very simple to understand. And second one is that those chemical compounds which are synthesized by the plants, fungus and microbes, which are other than primary metabolites, are called as secondary metabolites. Clear? Secondary metabolites. Once again, those chemical compounds which are synthesized by the plants, fungus and microbes, which are other than primary metabolites. I mean to say, when you take the several thousands of chemicals from the plant body, in that out of several thousands of compounds, just remove the sugars, that is polysaccharide carbohydrates, protein, lipid and nucleic acids. What remains in the plant body are considered as secondary metabolites. So those, I can tell you some of the few very common metabolites. Secondary metabolites are tannins, toxins, drugs, antibiotics, scent, flavonoids, yes or no? Flavonoids and and still many are there, yes or no? So these all different chemicals are simply come under. So one more very important one is pigments. So these are all simply considered as secondary metabolites. So our scientists have discovered several thousands of different secondary metabolites, but till now their functional aspects are known. Only some of the secondary metabolites and their functional aspects are known, but still many secondary metabolites are yet to be discovered, and along with their functional aspect. But some of the secondary metabolites are already discovered, but their functional aspect is unknown. Clear? So this is what our today's topic, that is nucleosides, nucleotides, and one more is primary and secondary metabolites. Clear? So, and one small homework is try to search some more metabolites, that is secondary metabolites. Clear? So, and this is enough for today's class, and remaining things we'll discuss in tomorrow's class. Thank you.